Good morning, afternoon, and evening. Welcome to the Pulmonary Compartmental Absorption and Transit Model Seminar presented today by John DiBella and James Mullin. A few housekeeping notes. For optimal video and audio connection, we highly recommend you close any additional web or streaming applications that may affect your internet bandwidth. We take your privacy rights seriously, and by attending this call or participating in the Q&A session, you are allowing us to contact you for follow-up. This call is being recorded for future playback, and today's course structure will have lecture and examples followed by a Q&A session. Please use your questions panel to submit your inquiry at any time. We know there are lots of ways to spend your time, and we appreciate you spending the next 60 minutes with us. Now to begin, we have a quick poll question for the audience. How would you classify your experience with GastroPlus software? We'll give you a few seconds to answer before we begin. All right, coming up on getting ready to close the poll. Thank you everyone for participating. It looks like we've got a fair mix here. And now it is my pleasure to introduce today's moderator, Mr. Dan O'Connor, Director of Business Development for Simulation Sciences. Take it away, Dan.
Thank you very much, Arlene, and welcome all to what we hope will be a very enjoyable 60-minute tour of the plus side of GastroPlus. A lot of times people aren't fully aware of the many capabilities that are built into the plus in GastroPlus. So over the next week, this coming week, we will be highlighting many of the additional dosage routes and capabilities that you may not be fully aware of. So we welcome you to this meeting. Just a couple of preliminary thoughts about Simulations Plus. About a month ago, we did add our newest acquisition, Lixoft, to the Simulations Plus family of companies and products. Many of you are already familiar with Lixoft being a leading provider of PK/PD modeling software, and also very, very much a presence in the academic world of PK/PD modeling. So we welcome Lixoft to the Simulations Plus family. Just a note for you that have attended us before, that we do spend an awful lot of our time, effort, and treasure on investing in the advancement of the tools that you're here to learn more about. The additional dosage routes are all products of our software investment. And just last year, we invested, I think it's something on the order of between 15 and 20% of revenue sales in software R&D. So we are committed to the growth and the additional capabilities that you have come to expect in our software platforms. And then lastly, before I introduce our two speakers and presenters this morning, I just wish to extend a genuine thank you to all of you for the success that you've brought to Simulations Plus. Well, obviously, we can't do this without you. And as recently as 4 days ago, we were added to the Standard & Poor's Small Cap 600, which is a significant milestone in the growth of a small public company, which we are. So again, thank you very much for helping us be the financially viable and stable company that warrants the inclusion in an S&P Small Cap 600. It is now my pleasure to introduce our co-presenters this morning, Jim Mullin and John DiBella. First with Jim. Jim is a senior principal scientist who did his graduate work at Washington State University, holds a degree in chemical engineering. And I mentioned to Jim, what is he doing in the pharmaceutical world? And he said he doesn't know. So I think that's a question we should ask him later in this presentation. But 15 years of experience in computational modeling with the pharmaceutical industry. He spent 10 years at Ben Research before joining Simulations Plus just about 8 years ago. And his co-presenter this morning is John DiBella, who did his research and graduate work in biomedical engineering at Case Western Reserve University in Cleveland, Ohio. John spent the entirety of his professional career, which I think is now officially 17 years, with Simulations Plus, headquartered in Lancaster, California. John is one of those rare individuals who's enjoyed great business success while at the same time maintaining his world-class scientific acumen. So it's my pleasure to introduce John and Jim. John, please take it away.
Dan and Arlene, both, thank you very much for the introduction and the kind words. Hello, everybody. Hope you all continue to be safe and productive during these times. And thank you very, very much for carving out time from your busy schedules to learn a little bit more about what we are doing as a company beyond oral absorption. As good as our scientists and our tools are at being able to predict future outcomes and activities, back 20 years ago when we were first working on GastroPlus, we didn't necessarily have the foresight to appreciate that at some point it would become much more than just an oral absorption modeling and simulation engine. But we've been very fortunate over the course of the last 10 plus years to enter into a number of different collaborations with companies to build similar mechanistic absorption or biopharmaceutics models to really address all of the major delivery sites and routes around the body. And I'm very excited about the series of lectures that we've put together this week, where we will dedicate time to each and every one of the major ones that we have available for you to use today. I know originally this particular session, day one of the series, was to focus on both pulmonary and oral cavity because I have been known to pontificate in the lectures that I host. It was decided that we would focus our energies today on pulmonary only, but on Friday during the open Q&A session, what we will do is give a few minutes to going through a background on what we're also simulating for drug products through the oral cavity route. So for today, what we'll be doing, and just let me switch over to the laser pointer, is focusing on pulmonary, things that are going to be inhaled through the mouth or through the nose. And what we're trying to push, not only with the pulmonary model, but with all of these non-oral delivery route systems that we have in GastroPlus today, is this idea that you can generate insights which you can't easily measure. So in all cases, similar to what many of you have become familiar with using the ACAT model in GastroPlus, here for the pulmonary system, we have a validated, mechanistic, physiologically based biopharmaceutics connected to PBPK model that allows us to predict both local and systemic exposure. And why is this important for you? Well, what we can do is we can start to apply these models to help with translational activities across species or even population groups to help guide clinical development activities and also establish safety risk. We've gotten quite a bit of interest in this pulmonary model, not only from pharmaceutical and generic drug companies, but also from the chemicals industry, the consumer goods industries, where we have to worry about potentially some of our excipients or other ingredients in household products, cleaners, and so forth, if those are inhaled in some way, shape, or form, what are our safety margins in terms of the exposure and the concentrations. We can also utilize these models to help with optimizing therapeutic dose and dose regimen by linking local concentrations within the lung compartments to the therapeutic, or in some cases, adverse effects. And one thing that I'll stress in a few minutes when we start talking about the model in more detail is that this pulmonary system is bidirectional, which means that we can start optimizing for dose or dose regimen utilizing target lung concentrations as our variable with products that are either going to be inhaled through the mouth and through the nose, or things that may be taken orally or applied topically or injected and may systemically circulate and be taken up into the lung. So we have this opportunity to be able to, again, optimize dose and dosing regimens through things directly inhaled into the lungs or taken up through the lungs by other routes. And then we can also apply these models to help us with formulation questions and more importantly, help us potentially establish drug product quality specs for these orally inhaled drug products. What is going to be our specification around particle size distributions, for example, which may be impacting deposition fractions? What would be the impact of these changes to the shapes of the particles, the presence of carrier particles and the sizes of those? Can we establish some sort of safe space for the products which we are developing? So the take-home message here is that we can really start utilizing the local results that are predicted within the lung to help drive global decisions. So we call the pulmonary model in GastroPlus the PCAT model, which stands for Pulmonary Compartmental Absorption and Transit Model. This is the current schematic and framework that was developed originally in a collaboration with our friends at GlaxoSmithKline. And the way in which we build up a model, which Jim will go through a little bit later during the examples, is really a three-step process. The first is to define the properties of the API or the chemical and utilize whatever formulation information we might have to calculate or set up deposition fractions. This would be very similar to the setups that we would use traditionally if we're trying to simulate products which are orally delivered. Once we have that core set of physical chemical properties in place and we have some formulation characteristics defined, we can then connect our systemic compartmental PK or PBPK model, utilizing data we might have which was dosed through IV routes, for example, or data that we might have which is from studies where drugs were delivered through non-pulmonary routes, and using that to help us define what would be the systemic volume of distribution and clearance parameters, which we can connect then to that local lung model. And with the combination of the physical chemical properties, the formulation information, and our definition of systemic PK, we can start to predict both local and systemic concentrations. And again, these concentrations can then be utilized to help us solve some of the questions that were posed on the previous slide. The framework or the foundation of this PCAT model that we have in GastroPlus today was based upon the compartmentalization of the lung as published and described through the ICRP 66 approach. So we have in the program today the nose and lung compartments, the pulmonary system, divided up into a maximum of 5 compartments. We would have the nasal passage or nose compartment, the area for the extra-thoracic region here, which would be comprised of the some portion of the nasal passage, the oral cavity, the larynx, and the pharynx. The thoracic region here, the bronchioles, and the deep lung tissue or the alveolar space. By default, we will have these 4 major compartments in place, and we will utilize some equations that we'll talk about coming up to help us calculate what percentages of that initial dose, which may be inhaled, are going to be deposited into these major regions. Optionally, you will have control over whether or not you want to include the nose, the nasal passage, if this is going to be something that's important for you to define in the model. Typically, products which would be applied through the nose, like nasal sprays, are where we would be considering this, this particular compartment. Otherwise, it doesn't play a significant role in our simulations. As far as the processes or the mechanisms that we have incorporated into these compartments, that's outlined here in this particular diagram. So for any vapors that we may have which are being inhaled, and this is a new functionality that we'll talk a little bit more about coming up, we would be using Henry's law constant to help us model the partitioning between the vapor phase and the mucus compartment. And then once we are in this mucus layer here, we're going to be modeling some of the same processes that we would for compounds which are orally administered in the luminal fluid. We would be taking into account the local pH and fluid volume considerations, along with some of the API or chemical properties, to start modeling any dissolution of the drug or chemical within this mucus layer space. If the dissolved concentration is higher than the solubilities which are being calculated, we may see some precipitation in play. And then we will start to model the absorption into the epithelial cell layer or into the intracellular space, which can be modeled through passive processes or considerations with transporters. And once we are in this tissue cell space environment, there are a number of different processes which could occur and which are interacting with each other simultaneously. There could be some local metabolism which is taking place. There could be some uptake into lymphatic circulation. One of the newer mechanisms that have been built into the software is lysosomal trapping within the lung environment. And so depending upon some of the physical chemical properties of the drug or chemical, we could see some binding to the lysosomes, which would be impacting some of the metabolism and or absorption rates. And then we have our equations and our models to then simulate what would be the uptake into the systemic circulation, which can be modeled through a passive process or through interactions with transporters. After some period of time, any material that is in the mucus layer that hasn't been absorbed into the tissue cells could transit out through the mucociliary clearance process into the next compartment, or once we get up into the extra-thoracic region, that material that's in the mucus layer would be swallowed into the GI tract. We would have the connections to the stomach environment and be able to model anything that would be absorbed across the gut. So everything that we see on this particular diagram here is occurring simultaneously and for each of the compartments that make up the lung model. Now, when we start getting into some of the practical considerations of using this model inside of GastroPlus, we're going to be going to the PCAT Model Interface, which is accessed through the modules menu. Number 9, I believe, is Additional Dosage Routes, and there's a dedicated submenu for the nasal pulmonary system. It's important here to emphasize that there is no equation writing, which is required to make use of this model, but you still have full flexibility to make changes and to customize it as you see fit. The program comes pre-built with default physiology descriptions for humans, and these human models are going to be scaled based upon your selection of gender and age. And then we have preclinical models in place for the rat, the dog, and now the mouse. So we make our selection. The program will pre-populate all of the physiological parameters for the various lung compartments, and we can then start running some of our simulations. We could begin at a very early stage of the process, just predicting what would be the local amounts and concentrations within the lung. Once we make that connection with a systemic compartmental PK or PBPK model, then we can start modeling the uptake or the absorption into the rest of the body and see what would be some of the concentrations in plasma or non-lung tissue spaces. And as I mentioned earlier, this model works in both directions. So you see here these blue lines that connect the thoracic and bronchiolar spaces as an example here to this diagram of the systemic PBPK model is bidirectional. So if I were dosing something through the mouth or through the nose, some inhaled products, I may have some drug amounts or concentrations in these spaces here and see some uptake into the systemic circulation, where I would then model the distribution and elimination. But on the flip side, I could dose something directly into the GI tract. I could take an oral product, a capsule or a tablet, or I could inject something directly into the muscle space as an example, and then see how it circulates to the lungs and is taken up into these subcompartments here. So we have this really unique and nice capability of being able to help design doses and formulations that are administered through the inhaled routes or non-inhaled routes. Some of the compound-specific and physiological parameters which are going to be important for the pulmonary simulations are outlined here in these diagrams on the left. And we're able, if I go back here to this previous slide, sorry, we can double-click on any of these boxes to pull up the forms that you see here. So again, you have this flexibility to make modifications to any of the inputs. From a compound, a drug or chemical specific perspective, we're going to require inputs for the permeabilities in each tissue. This is going to describe going from the mucus layer into the tissue cells, the systemic absorption rate from the tissue cells into the bloodstream. We would also have estimates for the lymphatic absorption as well. Optionally, we could define any enzyme or transport parameters and any binding terms to within the mucus or cell layers. The program will provide initial estimates for permeabilities and systemic and lymphatic absorption rate constants based upon some of the properties of the drug or the chemical that you're working on. These parameters could also be optimized or adjusted utilizing any in vivo PK data that you might have available. From a physiological perspective, how do we describe each of these lung compartments? some of the important parameters which will be set up for you for those species that were outlined on the previous slide: the surface area, the thickness of the mucus layer, the volume of the tissues, the mucociliary transit time, and a couple of newer parameters based upon our most recent collaborations include lysosomal fractions and gas volumes for vapors. And if we do decide, for example, to run some simulations in a species or a population group which is not provided by default, we can very easily come into this window here, modify with the best information that we have available, and save our own custom user-defined physiologies for the modeling and simulation activities. So we have a description and a setup of that core set of drug or chemical properties. We have a description of the physiology, the local lung environment. One of the other key considerations for accurate simulations for inhaled products would be those deposition fractions. What percentage of the dose that we originally inhale is going to be deposited into these different lung compartments. The program comes pre-built with several different methods, which are going to be dependent on some formulation properties: particle size distributions, particle shapes, densities of the drug or chemical material, also the presence of any carrier particles that we may have in our products. And so utilizing some of this formulation information, we then have choices within the program to use the ICRP66 method or one of the updated Finlay approaches to then help us calculate the deposition fractions. And these deposition fractions are then going to be multiplied by our initial doses that were administered to help us then describe the initial dose amount in each of the lung compartments. We can also utilize some measured data for deposition fractions, whether we've run some cascade impaction studies or done some additional experiments. We can utilize that kind of information and manually go into the program with whatever experimental deposition data we may have. So there are certainly options for utilizing the theoretical approaches based upon formulation characteristics, or utilizing any experimental data you have. And another, again, unique feature with the PCAT model in GastroPlus is the ability to also define carrier particles. If we have an interest in trying to deliver some of our active ingredients deeper into the lung space, down into the alveolar, for example, we may consider utilizing carrier particles, coating those carrier particles with our drug material. And what we are going to be doing is defining what would be a maximum cutoff value for the API or the drug or chemical particle size that will coat those carrier particles. And then any particles which are above that size, we'll use our normal deposition calculator selections to define the percentages of the dose going into each of those lung spaces. And then anything that attaches to the carrier, we'll be using a separate set of equations to help us then calculate what percentages of the dose of that material coating the carriers are going to be going into the lung tissue spaces. So we have this flexibility here to mechanistically model deposition fractions when we have carrier particles present. This is a screenshot here of the regional deposition calculation window, which is where we can select which type of deposition model we wish to use. Depending upon the model we select, there may be some additional options that we can choose. This is the area here where we can define whether or not there are any carrier particles which are present. Here's where we can define our drug or chemical particle size distribution information. Up here in this options menu, we have the ability to also modify some of the inputs that would be associated with the ICRP 66 or Finlay approaches. And so based upon what we define here, the program will then describe for us those initial deposition fractions. If we do select from this drop-down menu to use a user-defined option, which means we have some experimental or measured data available, these boxes would become enabled and we can just go directly into those boxes and type in our numbers. Here is the list of dosage form options that are currently available to select from on the main compound page of GastroPlus. So the area where you have historically selected whether you're simulating for IV doses, IR, solution, suspension, capsule, tablet doses. We have for the pulmonary model a long list of different options, and the letters PL indicating that these models are associated for products which are going to be inhaled directly in, through the nose or through the mouth. And so we can model these products, whether the material is administered in a solution or powder form, through the mouth, through the trachea, through the nose, or something which may be infused over some period of time. And then you can see that we've got now a new selection available, which is for volatile compounds, vapors. There's a new vapor inhalation selection that we can also use here. And this vapor inhalation option was developed through a collaboration grant that we still have in place with a large pharmaceutical company. This was something that we entered into last year. It's a 2-year funded collaboration project where we were making some more enhancements to the pulmonary system and also validating it with a much larger data set. The two primary new features that were added as part of this grant are the handling of volatile materials and vapors, which includes now new mechanisms for modeling the exchange of drug or chemicals at the lung fluid air interface, and also modeling exhalation in elimination processes. And then we've also introduced the new lysosomal trapping considerations in the model, which will have some impact on the estimations of deposition fraction calculations and also on the unbound percentages within the tissue cell environment. So just briefly, some of the new inputs for the vapor inhalation dosage form that's now available. We're going to have on the main PCAT window options for describing additionally the inhalation flow, which is separate from that value which we would be utilizing when calculating the deposition fractions. So inhalation rates or inhalation flows is an important parameter for the deposition fraction calculations. We've also now separately defined the inhalation flow for these vapor products. And then we have some models that we've developed to help us estimate diffusion coefficients for the vapors, and then Henry's law constant, which we're using to help model the partitioning between that lung fluid and air interface. So there are going to be some new parameters which are compound specific that are defined on the PCAT window. And then there are also going to be some new physiology terms which are set up for you for those species that we saw earlier. The vapor diffusion thickness, gas volume, And this slide here outlines some of the species-specific ventilation rates that we've defined by default inside of the PCAT model. You see some of those references down at the bottom of the slide here. So for humans, dogs, rats, and mice, we have now tidal volumes, breaths per minute, ventilation rates, which are now part of the models for simulating vapor inhalation. And you can see some special considerations, lots of variability in the values which have been reported for the rats and mice. And these serve as the basis for showing wide ranges of predicted concentrations. This variability would be something important to consider if we were doing some population simulations for a group of rats or mice. And then finally, the new inputs for lysosomal trapping. This is an example here of a lipophilic basic drug molecule, which has a pKa equal to 8 and a log P equal to 3. The program now has a new tab for the different lung compartments, which is defined as lysosome trapping. And what we're going to be able to see first on the physiology window are some of those new physiological considerations for the lysosomal fractions in the different lung compartments. And then on the lysosome trapping window here, we will see what would be the estimates of some of the partitioning between the tissue space and the lysosomal spaces. And that's going to be calculated based upon drug or chemical properties, along with some of the physiological conditions for the lung tissue, plasma, and red blood cell environments. And depending upon the drug or the compound that we are working on, we'll see some big differences in simulation results in terms of the concentrations of drug trapped in the lysosomes versus the drug that we would see free in the intracellular space. We often get the question about the reliability of this PCAT model in GastroPlus. And as mentioned, we've been very fortunate to have several different collaborations with pharmaceutical partners. We've also been very fortunate to work with other academic groups as well, where we've not only assisted in some of the modeling activities, but had a lot of this information published in the public domain. And so this is just a snapshot of some of the posters and peer-reviewed publications which have come out from a variety of different organizations describing the validation of the pulmonary model in GastroPlus for different types of drug molecules and also chemicals. There was a very good publication which had come out in 2018 from Dow Chemical, as an example, where they validated the pulmonary model in GastroPlus across a set of chemicals, utilizing nothing more than the machine learning estimates from those chemical structures. So in terms of having confidence in the predictions, I would be happy to point you to a variety of different publications and posters that have come out over the years describing the accuracy of the results. That's a little bit of background on this pulmonary model. As Jim gets prepared to take you through an example and case study, Arlene, I think it might be a good time to pop up another poll question.
Thanks, John. You've given us a lot to inhale. All right. My interest in using the GastroPlus PCAT model is please choose all that apply to you. We'll give you a few seconds while Jim is preparing his next set to answer the questions. Just a few more seconds here before we get started. Great. Looks like to support formulation development. All right, Jim, take it away.
Okay, everybody, it's good to be here today. I'm going to go through a quick tobramycin example in the next 15 minutes or so, and then we'll have some questions. The goal here is that we're going to utilize PCAT+ to provide systemic PK parameters for pulmonary modeling, build a model for the pulmosphere formulation for tobramycin, and then apply the model to predict a second formulation at a higher dose, so we can assess a nebulized formulation. So the first step I'm gonna undertake here is to build the PCAT+ model for systemic PK. So what I'm gonna do is navigate to the human record in the database. I'm gonna load the IPD file, and I'm gonna fit the parameters to the pharmacokinetic parameters to the tobramycin in human IV infusion data at 1.5 mg per kg. And then we'll export those parameters. So let's do that really quickly. Here in GastroPlus, I've already opened up the database for tobramycin here. We have 4 records, one for just the compound properties itself, and then a rat, and dog, and human IV record. We'll only really be dealing with the human predictions here for this example. Tobramycin obviously is a not a very lipophilic molecule. It's got a log P of -4.8. It's got very low permeability and very high solubility. If I click the pKa table here, you can see that the solubility, it's basically freely soluble in water because it's a basic compound. Yeah, you would expect based on this data here that the limiting mechanism is probably going to be absorption, right? Because we have high solubility, the dissolution will be pretty much immediate in the lung mucus, but again, it'll be absorbing more slowly because of the low permeability of this compound. So let's go to the human IV infusion input. And right now, there's no pharmacokinetic parameters here for this compound. So the easiest way to get parameters set up for this is to go to Modules, PCAT+, and click that, and open the IPD data file, which we've already created, which contains the concentration time data for the human IV infusion. So here, I've already created the file with the plasma concentration time data. I've set the dose, infusion time, and body weight for this study. And then I can click OK. And then simply all you have to do is click solve to get the PK parameters for this IV infusion. And automatically PCAT+ will determine what type of model best fits your data. In this case, it's a 2-compartment model. If you clicked on 3-compartment, you can see that the predictions are really no better. And so it's going to select the model that best fits with the least number of fitted parameters. When you're done with this, you can export the data. Here we don't have to worry much about first pass extraction, right? Because this is an inhaled formulation, so it's going to bypass the liver first pass extraction. So we'll click yes, and now we should have our parameters in our pharmacokinetics tab here. And if I go to simulation tab and click start, I can run a simulation, and I should have a good match to the experimental data. We'll then use this record as a sort of our template to make our pulmonary formulation records. So if we look back to the slides here, the next set of steps are we're going to copy the human IV infusion drug record. We're going to load in the concentration time profile. We're going to set the pulmonary administration to PL powder. And then we're going to enter the particle radius, and we'll look at simulating this compound. So if we go back to our database, we can go database copy drug record. We'll click yes because we just edited the parameters in the database and we want to save those. And we'll name this record human pulmosphere. And in this case, we want to uncheck copy support files with the record. This is because we don't want to copy the IV infusion concentration time profile, right? We want to enter in a new concentration profile specifically for the tobramycin. So I'm going to uncheck that. And now we have our new human pulmosphere record, I'm going to change our formulation to pulmonary. That's all you have to do to basically trigger the pulmonary module for loading. You basically just will select a pulmonary formulation, and then automatically it'll switch over to the pulmonary model. So you'll see under modules, additional dosage routes, the nasal pulmonary model will already be on at that point. We need to change the dose to 80. We need to change the dosing interval to 0 because this is just a single dose. We need to set the particle size. In this case, the particle size is 1.7, 0.85, and we'll use 3 bins for the simulation. And you can click OK. Of course, this data would be coming from your particle size analyzer. And I'm going to click Save. I think we also need to change the body weight for this study. I think it was at 80 kilograms, so we'll change the body weight to 80 kilograms. At this point, I think we have the formulation all set up. We need to import in the experimental data to compare our simulations against this. So I think I have a spreadsheet with the raw data from the study, so we can enter that information in. So if we navigate to the spreadsheet, human pulmosphere tab, there's the time concentration data that I can quickly just copy and then paste into GastroPlus, going to File, Load, OPD. This is where you load all your concentration time data from your experiments, basically. So we can paste that in there. Always check the units to make sure that they're correct. By default, it's micrograms per mil, so we're already set up correctly, but in case you're using some other units like nanograms per mil, you would need to set that in the units field here. You put in the body weight and dose, so 80 and 80 in this case. You can click redraw to view the graph, and then you can go File, Save. Click OK. So now we have our concentration time data, but we also need to apply our deposition fractions. So they had some either measured or in vitro data deposition fractions here that they provided. So we can go to our nasal pulmonary window here by going to the modules options. And then from within this window here, you can set all the pulmonary parameters. And here we can click on the deposition model. So here we could see you could select different deposition models. By default, it calculates based on the particle size what the deposition fractions are. Since we're using user-defined ones, we're going to overwrite that. But yeah, you can also predict them from the particle size. So we're going to add in 43.6, 9.3, 11.3, and 13.6, and then click OK, and then save. And at this point, we could run a simulation. I believe we have everything set up correctly the way we want it. I'm going to change the simulation length to 18 hours because I think that's the last data point in the experimental data. And you'll see that we have some over prediction. And at this point, you might ask yourself, well, what's causing that? And I kind of went through that already, right? We looked at the compound properties. It's a highly soluble molecule. The dissolution is not going to be rate limiting. Clearly, the permeability of this compound is very low. So in this case, we could do a PSA to figure out the sensitive variables here, but it's pretty obvious. So we don't really need to do that. We can just go straight to optimizing the systemic absorption rate constants for the pulmonary model to train this. And then we can use those same constants for a new formulation to prove that it's predictive. So to do that, I go to modules, additional dosage routes, nasal pulmonary on, and we need to double click on one of these tissue sections. It doesn't really matter which one. And then we can set a new systemic absorption rate constant to get closer to the experimental data. And I'm going to start with 7e to the -5. What you want to do here is double click on this text box. And what that will do is allow you to set those same parameters across all the different compartments. So you don't have to enter that information into every single compartment in the model. So then you can click OK and then save. And then this should be a better prediction, although it's not perfect yet. At this point, you could then go use the optimization module to better improve the prediction. So it looks like we have too much absorption in the upper lung, like the thoracic region, and we need more of the absorption in the deeper lung because it's, the rise time is much faster than we're predicting there. So the way you would do that is go to modules, optimization, select parameters and objective function, and then you can look at pulmonary compound and systemic absorption rate constants will be here on the right-hand side. And you can select all of those, click next. And then you could use the default objective function weight here. I actually found for this example that 1/y was a little bit better because it wasn't properly weighting some of the points around Cmax. So if you change to a weighting that weights the larger concentrations more evenly for this data set, it works a little bit better. And that's of course just a trial and error type of thing that you may need to do. So we can then run the optimization by clicking optimize, and it'll save the data. And this only will take a few iterations to get to a much better optimized solution. So you'll see fairly rapidly over here, the R squared for this formulation is going to go from 0.75, and we're already up to 0.81, and it should get up to around 0.93, I would think, here within about 40 iterations. So I'll only let it go for a short period of time because we don't have a lot of time to waste here. So we'll just give it a few more iterations. It's up to 0.86. 0.87, 0.88, 0.89. So this is probably good enough. We've probably improved our model. Now we're up to 0.9, so I'm going to stop the optimization. And then we're going to save these parameters to the database by just clicking yes to save those changes. Now if we run the simulation, we should get a little bit overall better prediction to the earlier time points. And so, yeah, so we've now trained our model against one data set. And now quickly, I'm just going to use it to predict a new formulation. So I'm going to go database, copy drug record, uncheck support files, and say human Toby for the nebulized formulation. And this is a higher dose, so I think it's 300 mg. If I go to my spreadsheet here, it's a 300 mg dose, same body weight, and it's a different formulation. It's a PL solution. We'll put in 300 mgs as the dose. We'll save that, and then we'll go to modules, and we'll adjust our deposition fractions to what they'd provided in the paper. So actually right here, so I'll go in and again edit those for this nebulized solution. Click OK, and then save. We won't adjust any of the systemic absorption rate constants, right? Because those should be fixed across drug records because it's a compound specific property now, right? So we don't want to then optimize that as well. Oh, I forgot one last thing. We need to import in this experimental data for the nebulized solution. So I'll go back to the spreadsheet and import the concentration time profile. So file, load, OPD, paste, save. I'll put the dose in there and the body weight. Save. Okay. And then we'll click start. So in this case, it's not a perfect prediction, but it's within the error bars. And so we've used the same parameters across two different doses and two different basically dosage forms. And we're able to do a reasonably good job predicting the concentration time profile for tobramycin. So this shows how you can use, one data set to sort of train your model and then use other data sets to then, compare against that model to see if it's, validate it and predict rather than sort of fitting all your data sets, right? You want to kind of use some data to train your model and then some data to test your model. And that's what we've done here for this tobramycin example. So I think that's all I had to go through. I thank you guys for your attention today, and we'll jump into the questions.
Thank you, John, Jim, and Dan, and everyone who participated in the Q&A session today. There were tons to get to, but we will reply to you via email or our LinkedIn channel. Join us tomorrow, Tuesday, June 2nd, with Jessica Spires as she tours us through the transdermal PCAT model. You can find more tutorials on our YouTube channel and by visiting our website at www.simulations-plus.com. This concludes our webinar today. We will see you tomorrow.